You need to do a lot of questions. You need to understand the topic, not just cramming, because if you're just cramming, you won't be able to interpret. If just in case there is a question they have tilted a bit, ne? What is DNA replication? What is DNA replication?
It's a code of life.
Code of life. Okay. What do you understand by DNA replication? She's saying that DNA replication is the process whereby DNA makes exact copy of itself. Do you agree with her? Okay, she's saying yeah. So DNA is a process by which DNA makes exact copies of itself. Where does it occur? Where does it occur? Where exactly do we find DNA? When does it occur? When does DNA replication occur? You see those questions I'm asking you? Those are the questions we're going to bring in exams, and then you check yourself, either you know them or you don't know them. If you don't know them, then it means that you need more emphasis on those areas where you have challenges. It occurs during interphase. If you look at meiosis, if you go to interphase, you will be able to see that DNA replication occurs during interphase. How does it occur? How does DNA replication occurs? Yes, when they ask you how does it occur, they're asking you describe the process of DNA replication. Yeah, can I have someone to describe for me? You know me, I like to teach as I'm asking. I don't like to drive alone. I get bored. Yeah, so when I'm driving, I ask you. That's why this is my music. My music by asking you questions so that I also get what? Entertained. Giving me a wrong answer doesn't mean that now if you keep quiet, I feel bad. But if you give me an answer, even if the answer is wrong, I get to know where to correct. But if you keep quiet, then I won't be able to know where to correct. You understand? Yeah, so it's better to give me an answer, at least what you have in the head, so that at least I can rectify that. Yes, my brother. Explain for me the process of DNA replication. To make a copy of itself. Yes, thank you. I saw the hand.
So talking about how DNA replication takes place.
Yes.
That would be summarized. And then the ones between the natural space they break. And then a template
Do you know? Do you put on the genes? Do you know a gene? No? How do you put it on? Up, bottom to up, ne? And then when you want to remove it, down, ne? Like this, ne? You don't tie anywhere or zip out somewhere. What happens? What happens when you zip up? It closes. What happens when you zip down? When you zip down, you see, when you zip, what forms? You form two sides, each is complementary to each other. You see, when you zip down, when you zip down, this side is not exactly the same as this side, but is compatible, isn't it? So that's exactly when DNA replication occurs. So when DNA replication occurs, this is what you're supposed to do. So you have DNA, you have DNA. Remember we say that. What is the natural shape of DNA? All those are questions we ask you in exam. What is the natural shape of DNA? Brainstorming. Natural shape of DNA? Two strands. Double helix. DNA is double helix. We don't want any other word. The only word you want to hear is double helix. Don't say helical. We don't want to hear those things. It's double helix. Is it clear? So now it's double helix. What does the word double helix mean? This thing is called helical. Isn't it? I have another helical. Since they are two, they are double. isn't it? Ne? Because they are forming that helical shape, therefore we describe it as double helix. Is it clear? So now when we say DNA double helix unwind, what happens? It means that some of you have, yes, your hair, if you look at the one you have made, ne, they are twisted. Ne? If you want to remove them, what do? You untwist them, isn't it? That's exactly what DNA does. So this double helix DNA, it unwinds. When it unwinds, it looks like that. Don't confuse the difference between unwinding and unzipping, because those are two different things. When you unwind, you are like this, you unwind. Ne? But the bonds between the two strands are still there. So you saying DNA double helix unwind, we give you 2 marks there. DNA double helix unwinds, you see? Depending on the marking guideline. You understand? Yes. So after that, you talk about the weak hydrogen bonds break. That's when you are busy pushing it down. You are unzipping. Do you understand? So when you unzip, it means that the bonds between the nitrogenous bases from one strand and another strand, they break. Is it clear? So we shall say that the weak hydrogen bonds break, or you can say that it unzips. It's the same thing. Ne? So it means that after that, you're going to find DNA looks like this. It looks like looks like that. You understand? Take an example that I have A, I have G, I have C, I have A, I have T, I have G, I have A, whatever. Ne? What do I have this side? A goes with T, C, G, T, A, ne? C, T. You see? So this strand is not exactly the same as this, but this strand is complementary to this. Do you understand now? Yes. DNA double helix, it unwinds. It looks like this. After unwinding, the weak hydrogen bonds break. It looks like this. What do you form when they break? What do you form? You form what? Two complementary strands. Okay. What do you form? 2 complementary strands. 2 templates. Okay, thank you. You form 2 separate. First say this. You form 2 separate strands. Don't say 2 strands. That word is separate because it could be 2 strands, but they are not separate. You understand? You form 2 separate strands. We want to see that word separate. If you don't put it there, we might not give you a mark. Ne? Yes. You form 2 separate strands. All right. Now, remember, I want to form DNA, of which DNA is supposed to have 2 strands, isn't it? It means that each strand must act as a template. I see. You form 2 separate strands. Now you say the word. Each strand is, that word each is a marking point. If you don't write it, we don't give you a mark. That word, I repeat, that word each is a what? Is a marking point. You form 2 separate strands, each acting as a template. Is it clear? When you form now 2 separate strands, each is acting as a template. What do you use to form these DNA? So, if you say template, what does the word template mean in layman's language? Because we will be talking about template. But what does it mean? Do you understand when I say template? What clicks into your mind?
One original strand.
You don't know how to explain it. But do you understand when I say template?
Yes.
What comes into your mind? Strand.
One original strand.
One original strand, eh? Okay. Girls, I think this is common. Do you check yourself in the mirror every day? What do you see in the mirror? Huh? You see yourself. Oh. But are you sure you're inside the mirror? It's a reflection, eh? Good, good. So the template will be there acting as a mirror. So a reflection of it is formed. Do you understand? That's the meaning. So it means that in layman, don't write it in exam like that. It's a mirror. I'm just explaining so that you understand better. You understand? And if you check in the mirror, it's not exactly the same, because when you close the left, it closes the right. You understand? So that's exact meaning that you, the picture in the mirror, and you are just complementary. Left leg, right leg. You understand? If you look at the shoe, they are the same, but they are not exactly the same, because one is left. If you put the left in the right, it won't fit, isn't it? So meaning that they are complementary. Do you understand now? So a template looks like a mirror. So now it means that each strand is going to act as a template, meaning that each strand is going to act as a mirror so that an image is formed there. Do you understand now? All right. Let's look at it. Remember, A goes with T, and then G goes with C. Cool. Now, a complementary strand is going to form on each template, isn't it? Whereby adenine is going to go with thymine, guanine is going to go with cytosine. What does it use? It uses free-floating DNA nucleotides. Listen carefully. Uses free-floating DNA nucleotide, because if it's DNA replication, we call it free-floating DNA nucleotide. If it is transcription, it uses free-floating RNA nucleotide. So what you are forming are the nucleotides which you use. Is it clear? So now let's show you. Let me show you something which I need you to see critically. So A goes with T, C goes with G, G goes with A goes with T, T goes with A, whatever, ne? C, and then this is T, isn't it? Look here, there is something which you need to understand, ne? Yes, because maybe you have been saying it, but you don't know where it's derived from. A goes with T, C, G, G with C, T, A, isn't it? T, G, and then A. Good. Now this one becomes a strand. Okay. It becomes a strand. You see? And then also this one becomes a strand. Isn't it? In other words, now I have one DNA and another DNA. Isn't it? Let me say DNA 1, then DNA 2. Ne? If you look at DNA 1, it has this strand. You see, it is original from up, I think. And then it also has this strand which is new. So it means that this DNA which is formed, it has one original strand and one new strand. I think it. Even this one, it will have one DNA strand which is original and one new strand. You see? So both, they will have one original strand and one new strand. Have you seen it? Then we say that these two new DNA, they are genetically identical. Are they identical? He's saying that these two new DNA are genetically identical. Are they identical? Yes. Someone is saying no, they are not identical. Let's find out. Let's find out. What do you have here? A. What do I have? I see. I see. And then this one? I see. Isn't it? Are they genetically identical? Yeah, they are genetically identical. So that's how DNA replication occurs. So you form two new DNA which are genetically identical. So in other words, if you are asked to describe the process of DNA replication, how does it occur? You see, it is a simple thing, ne? But don't make it complex for yourself. Ne? Yes. Basically, you say double helix DNA unwinds. It's a mark. Weak hydrogen bonds break. A mark. You understand? To form two separate strands. A mark. Each, that word each is acting as a template. A mark. Are you saying? Then you talk about that. Complementary strands are formed. You understand? A mark. Whereby adenine goes with thymine and guanine goes with cytosine. A mark. Are you seeing? Using free-floating DNA nucleotides from the nucleoplasm. A mark. You understand? So we said that when you use free-floating DNA nucleotides from the nucleoplasm, a complementary strand is what? Is formed, whereby adenine goes with thymine and guanine goes with cytosine. You understand? So you form two new DNA, each with one original strand and one new strand. Are you seeing? And then these two new DNA are genetically identical. They are genetically identical. Is it clear?